All right, hi everybody. Time for other blood group systems because you just got a handle on all the other blood group systems. So again, I'm going to skip through the objectives. You can utilize these to help organize your notes and follow along with the PowerPoint. Um, so the first blood group system we're going to talk about is the Kell blood group system. So I do want to clarify that these are basically just defining groups of antigens that are on your red blood cells. So the Kell system is similar to the Rh system, and they have two major antigens. So the big K and the little k, Chilano. So the big K, only about 9% or less of the population will actually have it. And you probably have heard this being called Kell. This is not the Kell antigen. It is the big K antigen, or K1 antigen. The little k, we usually will call it Chilano, or K2, and more than 90% of the population has this. So this is typically not one of our common antibodies that we have an issue with. So they are antithetical, and they are well developed at birth. The K1 or big K tends to be the most immunogenic, second to the D antigen itself. And one of the things that we can see is more commonly our common antibodies that we're going to identify include the big K antibody. And then the next would be the D. Typically speaking, this is something that we will give, and we'll typically see passive D, but we can see anti-D just because of its immunogenicity. And I forgot to, just to go back, I don't want to confuse anybody, but antithetical, this basically means that they are a pair of antigens that are coded by different alleles of a single gene. So basically what happens is we can inherit an allele from our parent that can code for big K or little k, but not both. So we get one from each parent. Next, we'll move along. We do have some other antigens that are in the Kell system, and they're analogous to the Rh system, like big C, big E, little e. So we have KPA and KPB. So KPA is a low frequency antigen, so only about 2% of the population would actually have it. So this is typically not an antibody that we would detect. We wouldn't detect an anti-KPA. And same going for KPB. It's a high frequency, so 99.9% of the population do have it. So typically speaking, if we do identify this as an antibody, it's an an autoantibody. Same thing with the JSA, JSB. So we have a low frequency, so typically in Caucasians it's 0.1 or 20% in blacks, and JSB is a high frequency, 80 to 100%. So when we're talking about antibody detection, and we'll talk about that next chapter, typically speaking, this is going to be where we look at the KP and the JS antigens, and we're typically not going to identify an antibody against these because they're low freq and high freq, or frequency, so we don't see those type of antibodies. So our Kell antigens, what's special about them is they have these disulfide bonded regions on their glycoprotein, so they are sensitive to sulfhydryl reagents. So this would be our 2-ME, DTT, and AET. So typically speaking, we would use reagents like that if we're trying to select out, but we'll talk more specifically about those when we talk about antibody identification. So you have K0 or Kell null phenotype, where they basically don't have a Kell system antigen at all, or they have this KX antigen. So what ends up happening is we can actually see if there is immune stimulation of these patients that are K0, they can actually develop what's called an anti-KU. So KU will be present on red cells that have KEL antigens. So it's similar to when you learn about hepatitis, and to have Hep D, you have to have Hep B. So if you don't have Hep B, you don't have Hep D. But so that's typical like that. So the KEL genetics, there's sets of alleles. So big K, little k, KPA, KPB, JSA, JSB, KEL11, and KEL17, which is our WKA. Your high incidence alleles are the little k, KPB, JSB, and your KEL11. Low incidence alleles are your big K, KPA, JSA, and KEL17. And I'll show you how this is expressed when we do antibody identification in a screen antigram. So our KEL antibodies themselves are immunoglobulin G, so IgG. They are stimulated by red cells, so this would be either by transfusion or pregnancy. They do agglutinate best in your indirect antiglobulin test or your antibody screen IgG phase. They don't usually bind complement. Doesn't say that they never do, but typically they're not going to. It will be associated with hemolytic transfusion reactions and HDFN. There's no effect when we treat them with enzymes. And then your anti-big K or K1 is the most common. Your KX blood group system, this is phenotypically related to the KEL system, but they are not genetically similar. So patients that have no KX antigen can actually demonstrate red cell abnormalities. They call this the McLeod phenotype. So we can see acanthocytes, reticulocytes, which means they'll be more permeable to water, and the red cell survival will decrease. So we will typically see this in males because it's inherited on the X chromosome. So the McLeod syndrome is basically the McLeod phenotype is attributed to the McLeod syndrome, where we'll have red cell abnormalities, muscular and neurological issues, and an increased creatine kinase or CK level. We can also see it associated with chronic granulomatous disease, which you probably learned already in immunology, where our phagocytosis is impaired. So our white cells will actually phagocytize bacteria and foreign entities, but we can't destroy it because we don't have the granules that we need. So then moving on to the next blood group system is the Duffy blood group system. So this blood group system we have at birth. They are destroyed by enzymes, which means if we pre-treat a panel with enzymes, it will make these antigens go away. It'll take them off the red cell. So your Duffy A and your Duffy B, they are co-dominant alleles, and they're most important for transfusion purposes. So our antibodies are your anti-Duffy A and anti-Duffy B. They are IgG. They don't bind complement. So this little sun means that they like the heat. They are stimulated by transfusion or pregnancy. It can potentially cause HDFN, but this is not one of the common ones that we see. And they don't, these antibodies do not react with enzyme treated red cell reagents because the enzymes destroy the Duffy A and Duffy B antigens that are on the red cells. So one of the things that's fun is most African Americans are Duffy A negative and Duffy B negative. And what's really cool is that this has kind of been postulated as protective evolution because Plasmodium knowlesi and Plasmodium vivax will actually not invade red cells that are Duffy A and Duffy B negative. So these antigens are basically receptors for that malarial merozoite to actually attach to the red cell. So it's almost like the African Americans, when they were developing, they developed this protective phenotype, which is really cool. So we really find this phenotype in West and Central Africa. So this is something that really kind of helps prove that there is selective evolution, and this is survival of the fittest at its greatest. So the kid blood group system is your JKA, JKB, and JK3. So in the US, most Black Americans, 51% are JKA positive, B negative, and Caucasians are JKA positive, B positive, or at least 50% of the time. JK3 is present whenever both JKA and JKB are present. So the kid null phenotypes, so JKA neg, B neg, are typically from the Far East. This was one of my certification exam questions, so remember that. And you could potentially see an anti-JK3 antibody, because again, JK3 they don't have, because they don't have JKA and JKB. So remember this. And these red cells are resistant to our 2M urea reagent. So our kid blood group system shows dosage and is enhanced by enzymes. So our antibodies, again, sun here, they like heat. So that's IgG phase, and liking 37 degrees Celsius is typical for our IgG phase antibodies. They are considered clinically significant. They can bind complement. They do cause hemolytic transfusion reactions and HDFN. They are the most common cause of delayed hemolytic transfusion reactions after transfusion, and they usually appear with other antibodies when detected. So that makes it real fun when we get a double antibody. I'll be scanning up an antibody packet for you guys once we start looking at that for you guys to work on for the rest of the semester. Detection of these is aided by enzymes, low ionic strength or LIS, and polyethylene glycol or our PEG. And we'll utilize enzymes on this to maybe get rid of some of the other antigens so we can help detect it. Our Lutheran blood group system. This is a pretty large blood group system. So there's 19 antigens. They're weakly expressed on cord blood cells. Most are high incidence. So antibodies to the Lutherans are incredibly rare. So our primary ones are LUA and LUB. So 94%, 92% are LUA negative, B positive. 7.4% are A positive, B positive, and 0.2 are LUA positive, B negative. And then the null phenotype is pretty rare, and it's inherited recessively. Enzymes have no effect on Lutheran. So our typical ones that we look at, LUA and LUB. So LUA, you notice we see cold and hot. So we can see them IgM in nature and IgG in nature. So the IgM will prefer the room temperature reactions, and the IgG will prefer the 37-degree reactions. So here if it's an IgM in nature, we're going to see it best at room temperature. Typically what we'll see is a mixed field pattern. This is not considered clinically significant. So LUB or anti-LUB is pretty rare because it's a high incidence antigen. It is IgG, hence the sun, and it reacts best in your IgG or AHG phase in your indirect anti-globulin test. It will also show mixed field, and we do see this associated with transfusion reactions. So if we rarely identify this, then this is actually considered clinically significant. So our Lewis blood group system, these are found in secretions and plasma. So when they're found in secretions, they're glycoproteins, and when they're found in plasma, they're glycolipids. These will absorb onto the red cell membrane. Typically speaking, most commonly, we're going to see LEA negative, B positive. Sometimes we can see LEA positive, B negative, and then the LEA negative, B negative is on the lower incidence, and then the A positive, B positive is pretty rare. And the reason being is these are not alleles. The Lewis system depends on our H, SE, and LE genes. So the little LE, little H, and little SE do not produce products. So if we inherit the LE gene, then we will produce the LE substance. So then if we are going to convert our LEA to LEB, we have to inherit the LE, H, and SE genes to convert LEA to LEB. So the LEA B positive red cells are pretty rare because once we get that LEA, we're going to convert it to LEB. So this is an example to show you what genes are present, because here we have all of these genes, okay? So we have all these antigens and secretions, which means we get to this end point of LEB, just to explain that. So it's almost like LEA goes away once we make B out of it. So our Lewis antibodies, these react, they're typically IgM in nature. They react at room temperature. They are produced by patients that are LEA negative, B negative. They're not considered clinically significant. We can see agglutination at any phase. Enzymes will enhance this reactivity. Anti-LEA will bind complement, so it could cause in vitro hemolysis. But again, it's not very common to see this. We can have neutralization, confirm the presence, or eliminate reactions with Lewis antibody here. So your I blood group system, or your little I antigens. So your I and little I are not antithetical antigens. They form from precursor A, B, and H chains on our red cells. So newborns will have the little I and adults will have the big I. So the little I is a linear antigen. So this will convert to the big I, which is a branched antigen, as our cell or our kids as we mature. So typically speaking, these I antibodies, or your big I, these are going to be our cold reacting. Usually they're considered an auto antibody. So this is very common for us to identify an auto I. So they do bind complement. They react at room temperature. They are IgM. Not clinically significant. An allo anti-I is really rare because most of us have this. We can pre-warm to get rid of reactions in an auto. So we often found these as a compound antibody. So they're found as anti-IH, and they'll have stronger agglutination with red cells that have a lot of H sites, like our O blood type or our A2 cells. So one of the things that's funky about this is our auto anti-I is often seen in Mycoplasma pneumoniae infections or cold hemagglutinin disease. And our anti-I, which is the allo antibody, we can see in infectious mononucleosis, lymphoproliferative diseases, and sometimes cold hemagglutinin disease. So our P1 PK blood group system, this is our P1 and our PK antigens. They are detected in plasma and hydatid cyst fluid. They're considered a globoside blood group system, or your P antigen, and then a globoside blood group collection, so your Luke and PX2 antigens, Luke as in like Luke Skywalker. P, PK, and Luke antigens are high frequency antigens, so that means most of us have them. So you'll notice that this chart kind of goes through the antigen characteristics, possible antibodies, and where do we care? Well, typically, the only one that we're really looking at as clinically significant is a P1K and a potential P. But again, it's pretty rare to see these phenotypes, so it's not very common to see these antibodies. So our anti-P1 we'll find in P2 individuals. They are IgM, enhanced by enzymes, non-red cell stimulated, and we can neutralize them. Our auto anti-P is typically with our paroxysmal hemoglobinuria. So it is IgG in nature, and we call it the Donath-Landsteiner antibody. So we can see this in kids after viral infections. And then your anti-PP1-PK, that's kind of fun, huh? It's in with that rare null phenotype. It can cause in vitro hemolysis, and if we do rarely find it, it is considered clinically significant. So then we move on to our MN and S blood group system. So Typically, they're M and N, and then S, little s, and U. So our M is coded by glycophorin A, our M and an N. S, little s, and U are glycophorin B. So our membrane structure is called sialoglycophorin A for M and N, has 131 amino acids. And then our membrane structure for S, little s, and U is sialoglycophorin B, hence on glycophorin B, and it has 72 amino acids. So M and N differ at positions 1 and 5, and S and little s at position 29. M and N show dosage where the homozygous inheritance enhances agglutination. So here, these are considered homozygous, which means the red cells have more on this cell, more M, and on this cell, N. So there's more, so it reacts better. The S and U antigen, we will inherit U when the S or little s is inherited. So our MN and S antibodies are all kind of connected. They tend to be in the same family, but again, pretty high frequency phenotypes. Sorry, lost my mouse. So then we actually have some other miscellaneous systems, anywhere from Diego to Cartwright, all the way down to SD. And I leave this chart in here. This is not something that we see very often, but you do need to know the names of these and what the antigens are. But typically speaking, this is going to be one of the things where maybe we've identified an antibody. We know it's there, but all of the other antibodies have been ruled out and we can't quite figure it out. So sometimes they will have extended antigen typing on some of the panels. And I can show you that once we start getting into that. So what you might want to do is listen to this now to learn it. You may want to come back and listen to this again after, or at least review it after we've gone over how to identify them. And I think what I'll probably do is try and do a screencast of me um, doing an antibody rule out. So I've got to figure out how I'm going to do that because you need to be able to see what I'm writing. So I'll let you know when I figure that out. Or we can do it live together if I can figure that. All right. So our HLA antigens, these are found on leukocytes and tissue cells. These, we will produce HLA antibodies if we've been transfused or pregnant. These have been associated with refractoriness and transfusion reactions. So one of the most common things that we see is platelet refractoriness, where we're transfusing a patient over and over and over again with platelets, but their platelet count just does not go up. So typically speaking, if that's the case and they still have active bleeding and you're giving them you're giving them platelets and they don't have idiopathic thrombocytopenia purpura or anything like that, we want to look to see if there's an HLA antibody associated with this. And I once had when I came in to work, I hadn't been at work for a couple weeks because I'm per diem, so I don't always work. And a patient had been getting transfused for like a week, multiple platelets, and it was just over and over again. This patient had a red cell antibody. Actually, I think he had two. So I questioned the platelets. So I pushed them to do um, at least an HLA cross match to see if we can find any cross matching. Well, when we did the HLA type, he had multiple HLA antibodies and he was incompatible with every single platelet that we tried unit in-house that we tried to cross match him with. So he was definitely a unique patient. So we need to make sure that we're looking at stuff like that because we can give him platelets till the cows come home. But what's going to happen is that his immune system is targeting it and chewing him up because he's got antibodies against it, which is causing cell destruction byproducts and therefore making him sicker. So we really need to be able to make sure that we pay attention to that. So HLA is like kind of like your backup plan or your second or third string, defense in football. So typically speaking, we're going to do HLA typing only if we suspect that there's some sort of HLA antibody, or we're doing hematopoietic stem cell treatments or organ transplants, excuse me. So we will inherit our HLA as part of our MHC. I think you probably remember this from immunology, where you have MHC class 1, class 2, and class 3 that are located on different cells. We will inherit one haplotype from each parent, and antigens are named with a letter following a number, so like A2. So remember your MHCs, so your class 1 are A, B, and C, your class 2 are DR, DQ, and DP, and then your class 3 will be your complement proteins and cytokines, and they're all on chromosome 6. So this is just an example of how you inherit. So mom and dad, and you can see, like, just if we look at child 1, for example, got the A2 from mom, A1 from dad, B7 from mom, B8 from dad, CW7 from CW7 from mom, CW3 from dad, DR17 from mom, DR4 from dad, DQ5 and from dad, and DQ2 from mom. So typical, just an inheritance where we get a copy from each. So HLA testing, we can have different methods of testing. So we can do what's called a cytotoxicity test method. So we use complement and dye to look to see if there's antigen antibody recognition. So cells that are positive will be dyed, and then that'll indicate a positive reaction. We can also, most of this, by the way, is done molecularly now. So the cytotoxicity is what we used to do. So we need to have matching HLAs for any type of solid organ or stem cell transplant. Patients can develop HLA if they're multiply transfused, if they've had a transplant, or if they've been pregnant. Typically speaking, we're going to see the HLA antibodies in transplant cases that were HLA unmatched, or multiple blood transfusions. So our stem cell transplants can be attained from bone marrow, peripheral blood, and cord blood, and we can treat all different diseases. But we need to make sure that we match at the allele level, so we can prevent rejection in graft versus host. Platelet antigens, so platelet proteins can elicit immune responses. So we can see post-transfusion purpura or neonatal alloimmune thrombocytopenia. So basically, in the neonatal, the newborn baby's platelets are getting destroyed by maternal antibody, because remember, antibodies cross the placenta unless they're IgG in nature. And then we can actually see destruction of platelets after a transfusion. So the most common one is HPA-1A. So this is different from HLA antigen. These are specific to platelet proteins. So these are the other blood group systems that we would look at. So these typically, so our RH, and our other blood group systems are the antibody, are the antigens that we are looking for antibodies against when we do an antibody screen. And we will talk more specifically about those when we start getting into antibody identification and things like that. So don't panic. This is a lot, but what I recommend is flashcards. Make a flashcard to say, okay, this is the M antibody, and then the characteristics of the M antibody or antigen. Is it clinically significant? Is it IgM, IgG, things like that. And that way, does it show dosage? Does it not show dosage? And we'll talk more specifically about dosage when we're doing the identification of antibodies or unknown antibodies. So hopefully this was helpful for you. And this will become second nature. Once you get out there and you do it and you see it, once we do an antibody screen that's positive and we do a panel, it'll start making a little more sense. And we'll get to it. So don't panic. All right. And please post any questions in the questions of the week that you want me to elaborate on. I've got lots of resources I can give you.